cancer has remained one of the leading causes of death worldwide for generations. Nearly 1 in 5 people will face a form of cancer during their lifetime, and tragically, almost 10 million lives are lost to this disease every single year. Pancreatic ductal adenocarcinoma or PDAC stands out with the lowest 5-year survival rate among all types of cancer, sitting at around 9 to 11%. The picture in Greece is even more sobering. Here, the incidence and mortality rates of PDAC are almost double the global average, with 7.9 new cases and 7.1 deaths per 100,000 people each year. On top of that, existing solutions like surgery are rarely an option due to the advanced stage and tumor location, while at the same time, other treatments offer little benefit and PDAC often shows strong resistance to them. This discouraging data led us to visit the Cancer Patient Association in Larissa, whose stories revealed the devastating impact of PDAC and the daily struggles patients endure. Inspired by their needs and together with experts in various fields such as molecular biology, biochemistry, pharmacology, virology, and computer sciences, we created Clotho, a targeted, personalized treatment designed to bring precision, reduce side effects, and most importantly, ease patient suffering. Clotho is a modular gene therapy delivered through an injection that contains a modified adeno-associated virus carrying a therapeutic transgene. Once the virus reaches the cancer cell, it produces 2 key components. First, circular RNAs that act as microRNA sponges, binding the targeted tumor-promoting microRNAs, thereby making cancer cells more sensitive to treatment. Secondly, the enzyme cytosine deaminase that will convert a subsequently administered product, 5-fluorocytosine, into the active chemotherapeutic agent 5-fluorouracil, which directly induces cancer cell death. Our solution's personalized aspect comes from our software Cathesis, a tool based on a genetic algorithm that receives as an input 1 to 3 tumor-promoting microRNAs of the patient and generates the optimal DNA sequence for a circular microRNA sponge. Clotho integrates modular gene therapy with computational design, offering a dual, targeted, and personalized strategy against PDAC.
Through our wet lab experiments, we could engineer the molecular machines towards bringing our therapy to life. We provided the biological insights needed to build our therapy and assembled genetic devices to test our hypothesis. In our 2-module system, a logical gate circuit controls the production of the Taf7 polymerase. The Taf7 polymerase is produced exclusively in pancreatic cancer cells and initiates the transcription of a bioactive microRNA sponge sequence. First, we combine the different regulatory elements to construct a novel and orthogonal system of transcription regulation for our synthetic microRNA sponge. Our regulatory module consists of the Taf7 polymerase gene. We placed it under the transcription control of the cancer-specific promoter HTERT, that is active only in rapidly dividing cells, and a microRNA response element for the tumor-suppressive microRNA LET7. To validate our choice, we constructed a model of differential equations to assess the system's behavior. And indeed, a regulatory module can successfully differentiate between cancer cells and healthy off-targets. At the same time, we strategically coupled the production of the ketosine deaminase enzyme with the Taf7 polymerase through the P2A linker, so that both enzymes are expressed only within the tumor. Our second module consists of a microRNA sponge sequence that traps the microRNAs responsible for tumor growth. The sponge consists of binding sites complementary to the microRNA targets, but not at the cleavage site of the risk complex. That way, the microRNAs are trapped and cannot promote cancer growth and chemoresistance. However, the problem we encountered with the microRNA sponges is that they are very susceptible to degradation. That is because the RNases of the cell digest the RNA from its 2 or 3 ends. We decided to counteract this problem by changing our microRNA sponge to a circular RNA form so that there are no free ends to assist with the degradation of the sponge. This is where our Star4 system comes in. To achieve the circularization of a synthetic RNA sponge, we use the Tornado system of RNA circularization. The system consists of two different ribosome sequences that take our sponge sequence as an insert. When that system is transcribed, the two ribosomes cleave themselves out of the RNA transcript, leaving the insert sequences with non-canonical ends. These ends are recognized by a ubiquitously expressed RNA ligase, the RTCP ligase, which joins the two ends and produces an RNA circle. To engineer the best possible sponge and by extension the optimal gene therapy, we needed robust characterization of the parts that compose our therapeutic transgene. Our attention turned to the thorough characterization of the Tornado system. In order to measure the system's efficiency in response to sponges' different possible lengths, we built Tornado devices using inserts of different lengths. To accurately measure the conversion from linear to circular RNA, we designed special primers for RT-qPCR that differentiate between the two forms of the insert. Our results provided viable insights to our system's function. By comparing the set of values between the circular RNA samples and the virus controls, we confirmed the circularization of 3 out of our 5 inserts. Moreover, we assessed the reliability of our results through analyzing the melting curves of our amplicons and managed to confirm the high quality for measurements. Finally, we used the set of values and the efficiency of the primers to calculate the conversion to circular RNA and construct a diagram comparing the different insert lengths to the efficiency of circularization. We see a clear increase in trend for the efficiency of circularization as the insert length increases. That grants great flexibility to the length of the microRNA sponge and confirms a core part of our therapy's molecular mechanism. Our characterization experiments are only the beginning of the incorporation of the Tornado system to the toolkit of synthetic biology. Through future in vivo experiments, we plan to open the way for this powerful system to revolutionize RNA therapeutics.
While designing our microRNA sponge, we expect to find an online tool that could combine binding sites for our 3 targets into a single optimized sequence. To our surprise, we couldn't find any that fit our needs. To fill this gap, we developed Clotho-Weaving, a software tool that generates synthetic circular RNA sponges for 1 to 3 microRNAs. It applies complementarity rules to design binding sites, then uses a genetic algorithm combined with RNA-fold to evolve candidate sequences. Each design is scored for accessibility and structural stability until optimal candidates are found. The resulting circular RNA sponge acts as a master inhibitor of cancer-promoting networks, regulated by its microRNA targets. While thanks to design, it can also be adapted to other types of cancer or even different diseases. By automating this process, Clotho-Weaving replaces repetitive manual work with systematic workflow. Its modular design allows researchers to quickly adapt sponges for different sets of microRNA targets, while its optimization engine reduces the design process from weeks of trial and error to minutes of computation.
Now, let's see how we plan to bring our project into the real world. When a patient gets diagnosed with PDAC, the doctors take a biopsy and send it to our university labs. There, our team extracts microRNAs and performs RNA extraction. Next, we analyze the data by using bioinformatics to identify the 3 most important upregulated microRNAs. By using our software Clotho-Weaving, we can design our therapeutic transgene cassette, and with collaboration with key suppliers, Clotho can be prepared and used for clinical treatment. Overall, Clotho can be administered by the medical staff very easily, while the patients benefit from the fact that it's not a multi-dose therapy. This means fewer hospital visits, making the treatment less tiring. And of course, every step of the process will strictly follow all necessary legislations, ethical guidelines, and safety regulations, ensuring that Clotho can move responsibly from the lab to the real-world clinical use. With Clotho, we're not just creating a cancer therapy, we're building an approach that makes a real difference for patients, families, and society. With the patient always in mind, we created a solution that reduces the severe side effects of traditional treatment options, giving patients more comfort, faster recovery, and more quality time with their loved ones. Additionally, improved precision means fewer hospital visits and less back and forth between clinical centers. But innovation is only meaningful if it is safe. That's why all the components of Clotho are based on technologies already validated in labs and animal models, with our next steps being to demonstrate their combined safety and efficacy. Of course, bringing a therapy to patients requires more than science. It requires a plan. That's why we've designed a clear entrepreneurial roadmap, beginning with proof of concept, followed by company formation and IP protection, then preclinical and clinical testing. Along the way, we'll build partnerships with suppliers, scale up manufacturing, and prepare for global distribution. To put this roadmap to the test, as well as strengthen our entrepreneurial skills, we participated in a bioinnovation competition. There, we pitched our idea to biotech experts, received valuable feedback, and connected with mentors. We also secured third place, which validated our belief that our vision can have a real-world impact. In short, our entrepreneurial journey shows how Clotho can grow from an innovative idea into a tangible reality. With a clear roadmap, strong partnerships, and a validated business model, we are confident that Clotho has the potential to make meaningful contributions to healthcare and society. Education and science communication are essential, not only for sharing knowledge, but also for building public trust, inspiring future scientists, and showing how research can directly address societal needs. That's why we shaped our education based into 3 goals: educating about synthetic biology, communicating our project Clotho, and inspire through the spirit of iGEM. To make our efforts meaningful, we followed a simple DBTL-like cycle: plan carefully, engage people through interactive and enjoyable activities, and then reflect on what worked and what could be improved. This way, every activity became a chance to grow and increase our impact. We started by visiting schools, where through presentations, experiments, crosswords, drawing, an obstacle course, and a mind map game, we introduced students to RNA, synthetic biology, and how an iGEM project is shaped. We also participated in several events, such as the 16th Electrical and Computer Engineering Student Conference, where we introduced the application of engineering in synthetic biology to students from dry lab-oriented sciences. At the Empower the SDGs event, we used a presentation and a Kahoot game to show how the sustainability goals can be applied in everyday life. Finally, at the Thessaly Tech Hub Forum, we engaged in discussions on sustainability and entrepreneurship with tech experts. Apart from this, we also organized our own events: a conference where we emphasized cancer research in molecular and computational fields by inviting experts as speakers, a workshop on drug dose modeling where participants learned to code in real time, and an event at our university with iGEM alumni that showcased real-world applications of synthetic biology and highlighted iGEM's role in driving innovation. Finally, we organized a webinar where experts and a former cancer patient emphasized the psychological dimension of treatment, highlighting how crucially good communication and trust between doctor and patient really are. Throughout the year, we also connected with other iGEM teams, sharing insights from our project and our overall experiences. Through these activities, we brought together people from diverse disciplines and backgrounds, showing that science extends beyond the lab. It thrives on collaboration, dialogue, and shared curiosity.
Although Clotho was initially designed for pancreatic cancer, its modular design means it doesn't have to stop there. By targeting microRNA dysregulation, our design can be adapted to other cancers and even non-cancer diseases. This scalability transforms Clotho from a single therapy into a platform technology, opening the door to treating multiple diseases with one adaptable system. Our journey has been about more than just science. We combined biology and technology to develop a therapy that prioritizes patient safety, well-being, and accessibility. Along the way, we built collaborations, learned from mentors, and engaged with the community, because innovation should not stay confined to the lab but be shared openly. Every patient matters. Guided by this vision, we work to create a modular, personalized therapy that is not only more efficient but also more humane. And while Clotho may be just the beginning, it sets the foundation for future therapies and hopefully inspires others to continue the effort.